Hi, my name's Doug Kelly, and I'm going to tell you about a new process discovered at the University of Rochester that we call antidispersion. But first, let me talk about what usually happens. Usually, a solute in a fluid spreads out and mixes over time. You can see it in this food coloring in a dish of water. You could see it in cream in a cup of coffee. You can imagine it happening when drugs are mixed into saline solutions. All three cases occur because solute molecules jitter around randomly and, given time enough, explore all the space allowed, thereby mixing. That's called diffusion, and it's a consequence of the second law of thermodynamics, which says disorder can never decrease. So you'll probably never see a solute do something like this when I play the movie backwards. On the other hand, you can speed up mixing by adding flow. For example, if you imagine some solute in a tube where there's flow along the tube, turns out from fluid dynamics, the flow is fastest near the center line, so the solute gets stretched out along the center line. But at the same time, molecules are diffusing, and so there's lateral spreading too, and the region with solute in it grows bigger and gets mixed in a lot faster than it would without the flow. This is a well-known phenomenon. You can simulate it as we did here. You can see in this movie that the region where there's solute gets bigger over time and less concentrated. The solute gets mixed in, and enhanced mixing due to flow is called dispersion. But we wondered what might happen if the walls of this tube were leaky, and leaky in a way that fluid could escape, but solute could not. So we built a model and we ran a simulation, and here's what we saw. Much to our surprise, the region where there's solute did not widen as it moved down the tube. It got narrower, and the concentration of the solute didn't drop, it increased. This solute got unmixed from the fluid. We hadn't seen that before. Nobody had seen that before, not in this context. And we were a little shocked because it seems at first to violate the second law of thermodynamics. Now, it doesn't really, because disorder is increasing a lot outside the tube. But within the tube, you can unmix the solute and order things very nicely. We call this phenomenon antidispersion. Once we'd seen it, we wondered if it might occur in nature and be useful to engineers. For example, around the blood vessels in your brain are perivascular spaces where water-like fluid flows and helps sweep away natural brain wastes. That's important because waste buildup is linked to diseases like Alzheimer's. Those spaces are known to be leaky, so we wonder if antidispersion might happen there. In drug manufacture and lots of other industrial processes, it's often the case that several solutes are dissolved in a fluid and you need to separate them. We wonder if antidispersion might be used to concentrate one and make it easier to extract from the other. If you're interested in learning more, you can read our paper, you can check out our website, and we are grateful for funding for this work from the National Institutes of Health and the National Science Foundation. Thanks a lot.